Today we are really excited to have Dr. Dahm present. He is a man that needs very little introduction in this group, so I will keep it relatively short. He completed his master's, Ph.D. and postdoc research at Oregon State. He studied a range of topics including nutrient biogeochemistry, the Columbia River input into the ocean, dissolved organic carbon in streams and rivers, ecology of old-growth forests, and effects of the Mount St. Helens eruptions on streams, lakes, and thermal springs. Throughout his career, his research has extended to Antarctica, Oregon, Washington State, New Mexico, Australia, France, Spain, Germany, and of course the Delta, where he now works as the Delta Stewardship Council Delta Science Program lead scientist. Among his numerous awards and accolades includes the recent 2016 Society for Freshwater Science Award of Excellence. And here's a happy picture of Cliff and his family accepting that award. And the very last thing I will say about Cliff is that he sure can come up with a talk title: Delta Nutrients, Sources, Sinks, Sags, Soups, and Sensors. So I usually, during these introductions, I like to throw up, a nice picture that represents the talk in one beautiful, wonderful picture. And he definitely did not make that easy for me, but I certainly tried. And here it is. And I think I got it all. I got the nutrients and the sources of the vegetables, the sink. The sag is the dead fish. That's kind of a stretch, but let's go with it. Soups and the sensor for the soup. And before we bring up Cliff, I just want to remind everyone that this talk is part of a series that we're having on the State of Bay Delta Science 2016. And the State of Bay Delta Science 2016 is a collection of papers that summarizes the scientific understanding of the Sacramento-San Joaquin Delta. It emphasizes the scientific progress made since the previous State of Bay Delta Science report published in 2008. And the batch of publications that includes Cliff's report on nutrients will be published later this year on the online journal San Francisco Estuary Watershed in Watershed Science. So please join me in welcoming Cliff.
Thanks, Nir. Creative picture. I'll try my best to hit all 5 of those, and actually I think I shall. I did want to say a couple things. So this is part of the State of Bay Delta Science series, and this is the 3rd one in the series. So the first one, as probably many of you remember, was given by Peter Moyle and Jason Baumsteiger, and it was about delta smelt. And that paper had been published in issue 2 of this year's San Francisco Estuary and Watershed Science. The second seminar was given last month by Michael Bettinger. Michael's paper is in press, accepted and in press, and was the topic then that he had about climate and climate dynamics, and particularly a focus on the drought. And so my talk is going to be about nutrients, and I wanted to comment, first of all, that this paper is submitted. It is not in press, in fact, and it's certainly not yet published. So I'm going to use a few bits and pieces from it, but this is really going to be an opportunity to talk a little bit about what's in the paper, but really it's going to be more of an attempt for me to weave some of my 40-plus years of work on nutrient biogeochemistry into maybe some ideas that might help us make progress on this topic as it involves working within the Delta. And I did want to comment that if any of you in the audience happen to be reviewers of this paper, be kind. Okay. I did want to say a little something also about the 2008 version of the State of Bay Delta Science. So the State of Bay Delta Science in 2008 was actually this hard copy book that was put out. There's a series of chapters in this. The book had no chapter on nutrients. Nutrients were not a topic. I spent some time reading through the 2 chapters that I thought might have something about nutrients, and that's the water quality chapter and the aquatic ecosystems chapter. You find the word nutrient, at least if I'm counting right, once in the whole document. So this is a topic that has not been covered in the previous version. I thought it was interesting in looking at some of the emerging topics that were listed at the end of the chapters on aquatic ecosystems and on water quality. Nutrients certainly weren't one of them. In fact, some of the things that they said were emerging issues that should be studied are fire retardants as a contaminant in the system. Another thing that they had that they thought would be the legacy effect of PCBs was an emerging topic. And the third one that they had were multiple mixtures of contaminants and their effects on fish species. So it's interesting to go back a few years and see what people thought of as emerging topics and then compare to what actually is coming out in the State of Bay Delta Science 2016. I want to do my acknowledgments up front here. You don't get up here and give a talk about nutrients in the Delta with as limited experience of working on nutrients in the Delta as I have without some really helpful co-authors. And the co-authors on this chapter are Annie Adelson and Maggie Crispin, who are in the audience, Brian Bergamaschi from the U.S. Geological Survey, and Alex Parker from the California State Maritime Academy. So those are the co-authors on this article. I also quite generously stole some slides from Ted Summer. I want to thank Ted for those. From Brian Pellerin, who used to be at the California Water Center, is now living on the East Coast, but very important in shaping some of the things that we know about the Delta through his very innovative work on sensor technology. And Matt Cohen from the University of Florida, who's done some really interesting work, which I think might be applicable to the Delta. All of my colleagues in the Lotic Intersite Nitrogen experiment, which I'm going to tell you a little bit about. It's a decade-long National Science Foundation-funded effort that I think really helped us understand the transport and fate and food web effects of ammonium and nitrate on aquatic systems. So I want to give you a little bit of background on that. The Australian Rivers Institute, where I did a sabbatical in 2007, who had some interesting ideas that I am promoting as ideas that might be worth doing here in the Delta. And finally, the Hydrogeoecology Group at the University of New Mexico, which is my home institution. So anyway, that's my introduction. Timeline there on the bottom are actually is sort of my timeline. So Nero already did that for you. I can throw in undergraduate work at Boise State University and working my way now to current position where I'm employed by the U.S. Geological Survey. So that's the chrono sequence. Here's my disclaimer, and this is a disclaimer that I wanted to start with. Nutrient biogeochemistry has been my research focus for over 40 years, and I'm very interested in this. It turns my crank. I really enjoy this topic, but I have not taken or analyzed a nutrient sample from the Delta. Any of you are actively taking samples? I'd like to go with you sometime and actually do that. But so far, I've yet to actually collect a sample from the Delta. My career in nutrient biogeochemistry started in 1973 at Oregon State University. Spent a lot of time at sea. About a year's worth of my life was spent at sea, mostly in the Antarctic Ocean, but also in the Pacific Ocean and the Atlantic Ocean. As time went on, I got increasingly interested in freshwater ecosystems, had the opportunity to work for quite a while at Crater Lake on the nutrient biogeochemistry of that system. We were able to show conclusively that Crater Lake is a nitrogen-limited system. In fact, there is less than a part per billion nitrate and ammonium in the upper 150 meters of that system. So very nutrient depauperate system, and one of the reasons why it's such a tremendously clear system. Also managed to spend a decade working at Mount St. Helens after it erupted in 1980. The picture in the upper right there, this picture, let me make this work. This picture here is something that our group was very proud of. It made the cover of Nature. So basically a paper. And then I increasingly decided that smaller streams and drier streams was my future, and so I began working in first old-growth forests in the Cascades, moved on to working on groundwater surface water interactions in the zone that we call the hyporheic zone, moved on from there to work a lot on high montane source waters in the in the southwestern United States, and most recently my research is focused actually on intermittent streams and rivers, which make up about 50% of the waterways worldwide, and I've been involved in a multinational study on intermittent systems. So that's my background. Now let's delve into some nutrient issues and some nutrient questions related to the delta. So this is the title of the paper that's under review at the journal. It's Nutrient Dynamics of the Delta, Effects on Primary Producers. And you can see the names of the co-authors, and you can see their institutions. And what I want to try to do in this talk is to actually go through those 5 S words that I used in the title. I want to talk a little bit about the sources of nutrients. Where do nutrients in the delta come from? I'd like to move on to sinks. Where do the nutrients in the delta go? I want to move on to SAGs, and when I'm talking about SAGs, I'm really talking about biogeochemical processes that occur that affect the solutes and the gases. So you can have a dissolved oxygen SAG, for example, or you can have a nutrient SAG, for example, when you have uptake of nutrients. And so I want to talk a little bit about those processes and how we might utilize some new information to analyze the importance of some of these processes. I want to talk a little bit to you about soups, and the soups are of 2 types that seem to be quite prevalent that we're talking about in the delta these days: harmful algal blooms and beneficial spring and now summer blooms that produce some of the kinds of base of the food web type of algae that we're trying to promote relative to stimulating some of the food webs that hopefully will make it up to fish. And then finally, I'm going to end up talking a little bit about some of the sensors and some of the new sensors that are being utilized around the world, certainly with a substantive amount of really innovative new sensors being utilized and deployed in the California Delta. So it's an area, I think, of really important progress. Okay, so let's start out with sources of nutrients. So when we were putting together this information, this was an opportunity for me to comb the literature, and with the help of my co-authors, there's actually some pretty nice papers out there that look at the sources of nutrients that come into the delta. So there's a number of nice publications, including the 2 that you see here, that actually look at the amount of nutrients that get into the delta. And the sources of nutrients are certainly scaled to the amount of water that these different systems put into the delta. From a quantitative perspective, most of the nutrients that enter the delta from the outside are coming through the Sacramento River system, even though the concentrations in the Sacramento are lower than the concentrations in the San Joaquin. The fact that there's so much water compared to the San Joaquin that the loads are generally, for most times of the year, the greatest from the Sacramento River. There are times of the year, however, that the San Joaquin can become important. The other interesting thing about the inputs is that there's a good timeline of information. It goes back decades. And so we have really good information for 30-plus years on the loading from those 2 major river systems. And one of the things that has come of these recent reports, and these reports have been, for example, part of the National Water Quality Assessment, NAQUA, is that in terms of nutrients entering the delta, things are actually getting better of late, not worse. So in terms of the amount of ammonium that's being loaded into the delta, or nitrate, or phosphate, tends to be, in general, some improvement. And that improvement seems to be linked to best management practices that are being done within the catchments. And so there's a lot of good information in this report on the right and in this paper that was recently published in this FUSE journal. So nutrients in terms of their external loading is well documented, and I think headed in the right direction. Within delta sources, we know much, much less about within delta sources. So one area clearly we don't know a lot about is the water that is used within the delta for agricultural purposes, and then some of that water is then pumped back out into delta waterways. That's a pretty big gap in our knowledge. Don't have a lot of information on that. We also don't have a lot of information on the sources of nutrients that might be coming out of sediments, for example, within delta with aquatic systems. So I think where our gaps are in terms of sources of nutrients are more within the delta questions than they are inputs from outside the delta. So once these nutrients are within the delta, what happens to them? What are the things that can occur to a nutrient once in the delta? Now this is something that there is certainly some good work here in the delta, but this is something where I thought I could link this well to this 10-year study that I was involved with. That was this national study called the Lotic Intersite Nitrogen Experiment. The Lotic Intersite Nitrogen Experiment, funded through the National Science Foundation in 2 phases, basically looked at the transport, fate, and food web effects of ammonium first, and then after 5 years of studying ammonium in catchments throughout North America, we then switched gears and did a similar kind of thing where we looked at the transport and fate of nitrate in these systems. For the ammonium experiments, they were 6-week continuous measurements of what happened to ammonium in stream and small river systems. For the nitrate study, it was actually 72 streams done over a 24-hour period. And what was innovative about these studies and what I think made them to be very highly published and very highly acknowledged as studies that really helped inform transport and fate and food web effects of nutrients was the fact that we used a true tracer. Now, what do I mean by a true tracer? A true tracer means that we didn't change the concentration of the nutrients in the system we were working. What we did was simply change the ratio of the stable isotope N15 to the stable isotope N14. So in natural systems, it's overwhelmingly N14 with a little bit of N15. What we did is just change that ratio. And you can change that ratio and have almost no effect on concentration. And so with the ammonium study, we changed that ratio so that we had much heavier ammonium, N15 enriched ammonium, and you could then follow that and what happened to the ammonium. And so this diagram here is an attempt then to show you how this actually works. So a tracer is added. It's enriched in N15. Number of things can happen to that ammonium. It can be transported through the system, and you can follow that, and you can watch then how much gets transported out of the system. It could be taken up by a variety of biological processes. So it could be taken up, for example, by aquatic plants. It could be taken up by benthic algae. It could be taken up by phytoplankton. It could be taken up by decomposing detritus. And so by sampling all of those compartments and making measurements, you can actually then follow where that ammonium is going within the system you're studying. And by doing this at a bunch of systems all over North America, we could then compare between the systems because we used exactly the same methodology at each site. And so we had a tireless postdoc who literally went from site to site and would spend a month at each site or 6 weeks at each site doing these experiments. And it really worked out very, very well. And the thing about stable isotopes is that the analytical methodology, which utilizes mass spectrometry, allows you to get very, very accurate measurements at a very, very inexpensive price. In fact, where did we do all of our N15 measurements? Stable isotope lab at UC Davis. So that was the place where all of these samples ultimately got ran. The studies with ammonium, I think, were very informative. One of the things, for example, that was found on a North American-wide scale was the importance of nitrification, the conversion of ammonium to nitrate by microbial processes. All systems, even with ammonium levels at near detection, had measurable nitrification rates. And it was a process that a lot of people had thought that the process would be outcompeted by biotic uptake. No, the chemosynthetic pathway of nitrification was actually important in all systems. So that's one example of some of the experiments that were done. The other thing that the ammonium tracer experiments did was allow us to basically look at both abiotic and biotic pathways. So with ammonium, it's positively charged. It can stick to a negatively charged clay. So how much sorption was going on in the system. In addition, ammonium in high pH environments becomes a gas, becomes ammonia, the gas, and that can volatilize out through the air, through the water atmosphere. So we were able to quantify that process. And so this was a methodology that I think worked well enough that NSF was willing to give us a few $1 million more to do exactly the same thing, only to use nitrate. And why the interest in nitrate? The interest in nitrate was because loading of nitrate into coastal ocean systems worldwide had become a serious problem. In particular, it was a serious problem in the Mississippi River drainage, and we needed to know whether or not small streams and rivers were effective in retaining or processing some of that nitrate. So that was the impetus behind the nitrate study. And so the studies basically used this N15 tracer approach. The method was developed at Woods Hole Oceanographic Institution back in the late 90s, early 2000s. And because of it, we were able to very sensitively measure these processes in these aquatic systems. And we started in small streams and small rivers. It's been extended now to some larger rivers. The cost goes up the more of the isotope you have to add. And actually now people are actually doing some similar kind of work in some coastal ocean and some estuarine systems. So that's the technique. And the other thing is that some very key metrics could be developed. And so let me just go through a series of slides here about a typical sampling. This is one of my doctoral students who did her Ph.D. actually on exactly this project. So she's out in the field adding this enriched isotope of nitrogen, in this case in the form of ammonium. This is the ammonium part of it. And if you take and sample moving downstream from the point of introduction, you'll find that the stable isotope begins to disappear from the water. And it can disappear because it gets absorbed, it can disappear because it gets volatilized, it can disappear because it goes into detrital pathways, it can disappear because it goes into algae or to aquatic plants. And you can sample all of those, you can figure out what that is. And what you can also get is an uptake rate. And so what you see there on the right is basically the natural log of this N15 tracer as a function of distance downstream. And one of the things that you can then get is an uptake length. So how far do you have to go downstream before this ammonium is taken up? And so for ammonium, these small streams and small rivers that we were working in, I don't think there was a site anywhere where the ammonium was not taken up on average in less than 1,000 meters. In most cases, it was less than 100 meters. So very effective processing of the ammonium. For the nitrate, it was longer. It stayed in the water longer. In some systems that were very nitrogen limited, those distances, again, were short in the tens to hundreds of meters. But in systems that had tons of nitrate, i.e. row crop agriculture in the Midwest, there was basically no uptake length. It was being transported through the system just like the water. It was basically a conservative solute. So these processes allowed us to measure a lot of key rates. Rates like nitrification, rates like denitrification, rates like uptake of this material into some of the key biotic components in a system. And so this is something that has some utility. In this case, we enriched the N15, but there's also the potential of using just natural abundances in certain systems. And so I am actively encouraging us to see whether that might work in some areas within the delta. And I'll come back to that at the end of the seminar. So what about nutrient sinks in the delta? So here is my opportunity to get up here in front of you and basically throw out an idea. This may be crazy, but I think it's worth considering. So here is a thought about nutrient sinks within the delta. So I remember my first trip to the delta when I arrived here in 2008. And I got this nice tour of the delta. And they were telling me, oh, the delta is a low productivity system. There's almost no primary production and slight limited, and yeah, it's really no nutrient story here. And I kept seeing all these aquatic plants everywhere in the delta. There were aquatic plants everywhere. It didn't look like a low productivity system to me. So I began to wonder about that, and here recently we've had a group of scientists come together in what's called the Delta Primary Production Workshop, and it was held in October 2015. The Delta Science Program helped support this effort, and we're talking about this kind of question. So let me throw out an idea to the group here. So there's been some very nice studies of annual phytoplankton primary production within the delta. So the JASB, Cole, and Claren paper is a very nice example of that. The number that is in that paper is about 70 grams of carbon per meter squared per year of phytoplankton production. And they say in that paper with a factor of about 5 between low and high values, okay? And 70 is the average. So low years it might be down 20 or 30, and high years it might be up maybe, over 100. So that's kind of the kind of numbers you're looking at. So that's phytoplankton primary production. Well, what about primary production for these aquatic plants that are out there? Well, there's two groups of aquatic plants that dominate the delta, certainly at least the delta waterways. One of those are submerged aquatic macrophytes, in particular Egeria densa, Brazilian waterweed. Well, I went out and started to try to find information on rates of primary production for Brazilian waterweed. I found some Egeria data, but I did not find any Egeria densa data. I did not do a rigorous data check. But what I did find is in Wetzel's textbook a nice summary of submerged aquatic macrophyte primary production worldwide. And the numbers that he gave for places where nutrients are in abundance were anywhere from 200 to 1,500 grams of carbon per meter squared per year for Egeria densa, or for the submerged aquatic macrophyte, of which Egeria densa is an example. So that's kind of the dynamic range that he's found worldwide for these kind of submerged aquatic macrophytes. The other group of organisms are the free-floating forms that dominate, and particularly water hyacinth. I think all of us know water hyacinth is a highly productive plant, and in fact, the rates of production in Wetzel's book for water hyacinth range from 1,500 to 4,400 grams of carbon per meter squared per year. So Ted Sommer, in a presentation of the IEP recently, pointed out that in 2015, about 30% of the waterways in the delta were covered with aquatic macrophytes, mostly invasive aquatic macrophytes. So these rates of primary production, if verified in the delta, and then linked to the elemental stoichiometry of how much nitrogen and how much phosphorus are in these plants, could give us a pretty good idea of the demand for nitrogen and the demand for phosphorus from this group of organisms. And my initial calculations would suggest that when we have these big water weed problems that we've been having of late, I think there's as much nutrient going into those plants as there is going into phytoplankton, and maybe more. So I think that is a worthwhile endeavor to begin to think about and potentially analyze. And what would you need to do? You would need to collect these plants in various places around the delta. You'd need to measure the carbon, nitrogen, and phosphorus content of these plants, and begin to look at the potential for these to be one of the major sinks. You could also use some of the new sensor data, and I'm going to talk about that later on in the talk. But this Delta Primary Production Workshop that we had in October of 2015, second workshop is in the planning stages, but this is the kind of question that we may be looking at is, where does the nutrients go in the delta that promote primary reduction? Is it mostly into phytoplankton? Is it mostly into submerged aquatic macrophytes? Is it mostly into the free-floating aquatic macrophytes? I think we can do a more quantitative estimate of this. With the free-floating forms, you know they're sourcing it out of the water. With the Egeria, with the rooted submerged aquatic macrophytes, there's always this question of how much is coming from the sediment versus how much is coming from the water, and that would also have to be determined. Okay, and this is the map that Ted nicely shared with me. This is Frank's tract in November of 2014 here. Work in 2014. This is Frank's tract in September of 2015. So with this kind of coverage of aquatic macrophytes, there's got to be a lot of nutrient going into these systems, and I think this is something worth doing research on and getting better numbers concerning how much nutrient is being sourced out of the water. Okay, so that's my sources and sinks part of my seminar. Let's move on to the soups part of the seminar. The soups part of the seminar, again, Ted Summer and colleagues, thank you for the slides. We've had some interesting phytoplankton dynamics in the delta this year, and some of that is desirable, and some of this is less desirable. And so I just wanted to go through some data from this year. That's recent information about the effect of nutrient loading into the North Delta out of the Yolo bypass. In March, probably most of you remember, the Yolo bypass spilled. We had a lot of water moving through the Yolo bypass. The Yolo bypass then began to drain as we went through later on into April. Began to see a pretty large bloom. That large bloom extended well into May. The bloom was made up of a diatom, and the diatom is Algalcyra. Some of you may remember it when it was called Melicyra. And this is an organism that had a very extensive bloom in the northern part of the delta for a few weeks to at least to a month. And so the people scrambled to get researchers out to the site, and so quite a bit of information has been collected. There's a lot of continuous sensor data that I'll show you in a little bit, but there is also, there's quite a bit of nutrient data collected, and that nutrient data is being analyzed, and it'll be very interesting to see what form of nitrogen was deriving this bloom. Is it a nitrate-driven bloom or an ammonium-driven bloom? Or was the ammonium, did we need to see the ammonium down to a low level before the nitrate started to be utilized? So I think we have an opportunity to look at some of the dynamics of bloom formation and nutrient biogeochemistry as this process proceeded within the delta. And then experimentally, a similar kind of study was done in summer 2016. This was a collaborative effort between many, many individuals who made this happen and organizations. And this occurred then in July. And that experiment was also nicely designed into a sampling protocol and a sampling scheme. So we had a number of good measurement sites. A lot of sensor data was being collected throughout this movement of water through the lower Yolo bypass into the North Delta. A bloom again occurred. This is the conditions. This is chlorophyll data. Find my cursor. This is chlorophyll data. This is chlorophyll down at 0 to 5 micrograms chlorophyll A per liter. This is pre-bloom conditions, and then you can see the starting and the development of this bloom that again lasted for multiple weeks. In this case, it's in August. Same organism as the bloom that occurred earlier from the natural spill of the Yolo bypass that occurred in the late spring, early summer. So very interesting that we can potentially manage the system for this kind of primary production. And the next big question, and I think an important question that a lot of people are interested in asking, is what effects did this bloom have on up the food web? So how much of this material moved into, say, zooplankton communities, and did it get up into an effect actually on some of the fish communities? So I think exciting and well-designed research and potential some important implications moving forward as we think about how to manage places like the Yola bypass. Now let's move on to our other soup, and the other soup is not as desirable, and that's the harmful algal blooms that have been problematic worldwide. Lots of reports this year about the problems nationwide, and California certainly has not been spared from this. Lots of problems here in California. Probably the one that's got the most national press is the one in Florida, but certainly there's been some major problems. A number of different harmful algal blooms are involved, but the one that seems to be dominant in many sites is Microcystis. And so Microcystis is showing up in a number of locations throughout California, including the Delta, and concern with algal blooms, these kind of harmful algal blooms are not only the ecological effects of these blooms on the food web, but also there's a direct human health issue that has to be addressed. Variety of toxins produced by these harmful algal blooms, including some pretty nasty neurotoxins, and certainly we've been seeing at some of our locations here in and around the Delta levels of cyanotoxins that are of significant concern. The Delta effects of Microcystis, the worst blooms this year have been in the South Delta. There's some very clear water quality effects that come with these blooms, including large swings in pH and dissolved oxygen in some of the channels. So there's another one of your SAGs. And then also ammonium has been shown to be the preferred nitrogen source for these harmful algal blooms. So this issue of ammonium interacting with harmful algal blooms is another area I think deserving some attention. Okay, another SAG. So this is one some of you probably heard me talk about this when I gave a short talk at the Interagency Ecological Programs Workshop. This is a paper that I make all of my graduate students read early on in their careers. This is a paper that was published by Howard T. Odom in 1956 in the inaugural issue of Limnology and Oceanography. This is a paper where Howard T. Odom basically pointed out that if you make regular, repeated measurements of dissolved oxygen and couple that with some information on the light field and the temperature and the barometric pressure, you can do calculations that would allow you to estimate rates of primary production in the aquatic system that you're studying. And there's one other important kicker in this, and it's the one that is the biggest challenge in using this method, and that is you also need to know something about the exchange of oxygen between the water and the atmosphere. And that exchange process is called a reaeration coefficient. There are some direct ways of measuring it, and there are some model-based mechanisms of estimating it. And so most commonly now people are using these model-based tools to estimate reaeration, but most of the good studies will also couple it with direct measurements to make sure that the model are well calibrated to it. So this is a paper written a long time ago by a very smart man who basically said, hey, if you make these kind of measurements and you get some of the ancillary data that goes along with these kind of measurements, you can estimate not only rates of primary production, you can also estimate how much respiratory activity is going. In other words, how much oxygen demand is in the system, chemical and biological. So the method is a very nice one set up. We've added a few nice little additions to this as time has gone on. We basically now are able to measure dissolved oxygen continuously with all of our new optically based sensors. So there's methodology to do like you see here, which is some work from Walter Dodds at Kansas State University, who was part of the Lotic Intersite Nitrogen this diet. All right. You hear it? This one has the lovely echo. Okay, so this is the method, but what we do now is we add continuous measurements of oxygen. So we do the oxygen better than we used to do. People are using this around the country, and the one person that I've had a great opportunity to work with closely on this is Matt Cohen. He's at the University of Florida. He's been making these kind of measurements in large rivers in Florida, and basically he utilizes these new generation of sensors that are able to measure continuously a lot of these variables, and he's added another wrinkle, and it's an important wrinkle. So not only are you able to measure rates of primary production and rates of respiration, you can also measure rates of nutrient uptake, and I'm going to explain how that works really quickly. So basically, he and Jim Heffernan at Duke have developed a method that uses daily SAGs in nutrients linked to the daily SAGs in oxygen to figure out how much nutrient uptake is occurring within an aquatic system on a daily time step. So in other words, the amount of milligrams of nitrogen per day or the amount of milligrams of phosphorus per day. And so what they've done basically is they have utilized sensors that can continuously measure nutrients, coupled that to sensors that continuously measure oxygen, and by looking then at the day-night cycle, and you look at the drawdown during the day and the build back up at night, you can use that information to actually generate rates of primary production on the x-axis and rates of nutrient uptake on the y-axis. And you can actually do a regression line on that, and you can actually look at the ratio at which these nutrients are being taken up. This is an example with nitrogen. Same thing could be done with phosphorus. And it's interesting, the molar ratio, carbon to nitrogen, in this study in this Florida river, had a carbon to nitrogen ratio of 24. Some of you may have taken ecology or oceanography classes where you learned about the Redfield ratio. The Redfield ratio is the ratio of carbon to nitrogen to phosphorus in phytoplankton in the world's oceans, and it's remarkably constant throughout all the oceans of the world. Works well in big lakes also. That carbon to nitrogen to phosphorus ratio in the Redfield ratio is 106 moles to 106 moles of carbon, 16 moles of nitrogen, 1 mole of phosphorus. So if you have a system dominated by phytoplankton, that carbon to nitrogen ratio should be 7, or approximately 7. This one's 24. What does that tell you? Lots of aquatic plants in this system. This is a system that has most of its production going to aquatic plants. Wouldn't that be interesting to know in the delta, where phytoplankton is dominant versus where some of these aquatic plants is dominant? Here's the method that works. All right, I'm going to end then with a couple slides on sensors. So back about 10 years ago, I started to get interested in sensors. One of my fellow graduate students from when I was in oceanography went on to be a sensor guru at MBARI, at the Monterey Bay Aquarium and Research Institute. He's been developing sensors for the oceans, and people working in freshwater have begun to embrace these type of sensors, and probably the group that led the charge here in the United States was the California Water Center of the U.S. Geological Survey. So here's some examples of some of these sensors. There's the SON sensors. They've been around for a long time, but there's also continuous sensors for measuring phosphate, continuous sensors for measuring nitrate. This table is some examples of some of the other things that are now either developed or in development stages. So there are sensors, for example, that are able to measure algal pigments. There are sensors that can measure some of the organic carbon components of water, and there's also some that are involved with measuring different aspects of the sediment composition and sediment types that are there. So these kinds of sensors are now getting more and more play. This is I'm sure some of you have seen this. This is a pair of slides that Brian Pellerin provided to me. They're from the San Joaquin River here in California. I gave a plenary talk in Berlin a couple years ago where I had all this wonderful information that I thought they wanted to hear about, and after my talk, all they wanted were these 2 slides. But this is an example of taking grab samples in an aquatic system every few hours, and in this case, they measured nitrate, and it was over a 3-day period. If you looked at this data, you'd say, okay, concentrations of nitrate are increasing, maybe 20% over that 3-day period. This would be the, probably what you would report out if you would analyze these data. But when you stick a continuous sensor in the system, this is what was going on in that same time frame. So here's continuous data. 2 things to note. One is that the continuous sensors agree very, very well with the lab analyzed data. And number 2, there's a whole lot more structure and a whole lot more going on than just the grab samples would allow. So these sensors, I think, are really helping us understand nutrients everywhere. And we're lucky we have a lot of these sensors deployed within the California Delta. And in fact, this data, to me, is impressive that they were able to do this and maintain this. This is a 13 to 14-month continuous measurement of discharge and nitrate at 2 locations in the Delta, one at Freeport, one at Walnut Grove. The black line that you see there is the tidally averaged flow, and then you can see the green and yellow are the 2 stations along the Sacramento River. A couple things jump out at you when you see this. First one is the very important role that big flow events play. This is from 2013 to 2014 data. So these flows are up in the order of 30,000 CFS, so not spill-type flows that would get water into the Yolo bypass currently. But you can see that the nutrients respond quite strongly to these flow events, and nitrate in particular is enriched. So it's probably not surprising that when you get flow events that spill into the Yolo bypass, you're probably putting quite a bit of nutrients into that system. And how those are processed and how they're utilized, I think, is another area well worth considering as a major research area. It also helps to explain some of the phenomenal productivity that connecting these floodplains with the river seem to generate. And then finally, my last slide. This is a slide from the Australian Rivers Institute. This is a slide that I also showed at the IEP workshop. This is a slide from a study that was done in Moreton Bay and the rivers that drain into Moreton Bay in the area around Brisbane, Australia. This is a program that I went to learn about when I was on sabbatical in 2007. The Australian Rivers Institute was the lead in developing, for what I have seen worldwide, one of the very best monitoring programs done anywhere to basically help make decisions on adaptive management and experimentation to deal with deteriorating quality in Moreton Bay. And so the initial program focused on the bay. After a couple of years of that, it became clear that they better start measuring what's going on in the riverine inputs to the bay, and so they expanded that. And they've used that then to make decisions how to invest finite resources in various kinds of aspects to try to improve the health of aquatic ecosystems in this area around in southeast Queensland. It's called the Healthy Waterways Partnership, or the Healthy Waterways Initiative. And one of the things that they did early on was try to assess the source of nutrients getting into Moreton Bay. And so they did some interesting studies, and one of those interesting studies was to use stable isotopes again. And so it turned out that the ammonium that is part of the effluent being discharged by some of the wastewater treatment plants in this part of Australia is enriched in the heavy isotope, and it provided a natural tracer, and that natural tracer then moved into various parts of the food web. And what you see here in these series of diagrams is the condition that existed in 1998. The areas in red are areas that have substantive effluent sourced nitrogen. And what they used actually to look for this were either macroalgae or aquatic plants, both of which we could utilize similarly. And this is what they found. So it was pretty clear that they had some point sources that were important. They invested $400 million in upgrading those plants. And as you can see, during the plant upgrades and then continuing to monitor, I can get this back. Here's 2001, 2002, and then 2008, that they largely were able to eliminate this source of effluent-derived nitrogen from the Moreton Bay ecosystem. There's still a little bit right down there, but this was a way of showing the efficacy of the upgrade in improving the condition relative to nutrient availability for the primary producers within this aquatic system. So again, this is something that I think might have some applicability here in the California Delta, and we have a major experiment that's being done for us as the regional sand facility is upgraded with partial upgrade due to occur in 2019 and complete upgrade in 2021. These are things that if we do the right kind of experimentation and measurements beforehand, might prove as a good mechanism to look at the effectiveness after they upgrade and see if some of the hypotheses that various people are putting forward actually come to be true. And so that's one of my little pet projects I'm going to work on up until I really retire, 6 to 9 months from now, or maybe a year from now, we'll see. All right, I'm done. I'm open to questions. I know I took most of the time, but hopefully you found this of some interest, and I'd be happy to address questions. I will repeat the questions after they're asked. By the way, my last slide is for you to read. These are my suggestions for future studies in the nutrients in the Delta. Yeah, question.
Yeah, you mentioned nitrate sensors being valuable to you. How valuable are they without also adding an ammonia sensor at the same time? I've heard that the ammonia sensors are not as reliable. There's how much biosettling and what the issue is, and look into it. But is there a lot of utility in just doing nitrate by itself?
The question has to do with the nitrate sensors. The nitrate sensors are UV-based, and they work very well, and they're very reliable. Yes, you are exactly right. If you're in a system where there's a lot of ammonium, and in addition to the nitrate, then you need both of the inorganic forms of nitrogen. The ammonium sensors are not as far along. They also are not able to detect at some of the low levels of some of the natural sources. So we've tried ammonium sensors back in New Mexico where I work. We can't get low enough detection limits for them. Here in the Delta, those levels are higher. The U.S. Geological Survey Group is working on ammonium sensors. They have had some successful short-term deployments, but nothing like the long-term deployment that you just saw for the nitrate sensor. If you have significant amounts of ammonium and nitrate in the system you're studying, you need to get both. You need information on both if you're going to do some of these uptake dynamics and uptake rates. So yes, they're not as far along, but there are a lot of smart people working on making them better. And I think that they will get there eventually. But part of it in some systems is always going to be a detection limit problem. So.
Any other questions?
Number second bullet up there, can you do it without models?
That's exactly what it's all about, is linking hydrodynamic models with biogeochemical models, yeah. I probably should have put modeling in there, but not being a modeler, I tend to forget that word.
In Australia, when they reduced their nitrogen loads, what happened to the macrophytes?
Actually, the way that those studies were done was they actually took and put planted macrophytes in various sampling locations, and actually then came back a couple months later to see what level of stable isotope had accumulated within the system, and they did studies to know how long it would take for it to reach equilibrium. What they were actually trying to do in Moreton Bay was get the macrophytes back. So the macrophytes are problematic. They had sediment erosion problems from their agricultural catchments, and the sediment was reducing light availability, and they had lost a lot of their desirable macrophytes that were nursery areas within the bay. So their whole goal was just the opposite of what we've got here. They were trying to get the macrophytes back, and they have been largely successful in doing that by improving riparian condition in the 4 catchments that were the major source areas for sediment. So. But what they did was use the macrophytes as one way of telling them how much of the nitrogen that was in these plants was derived from effluent, and that's what that diagram is trying to show. Other questions?